The overall goal of the following experiment is to identify interactions between different microbes that are mediated by secreted compounds and lead to changes in bacterial gene expression. This is achieved by co-culturing a bacterial strain containing a fluorescent transcriptional reporter with environmental microbes as colonies on agar petri plates. The orange circles represent colonies of the environmental microbes, while the blue circles are colonies of the reporter. Growth on solid media allows the metabolites that these colonies produce to diffuse through the agar and activate gene expression in nearby reporter colonies. The potential hits, indicated here with stars, are those colonies that may be secreting compounds that alter gene expression in the reporter. These organisms are isolated from the co-culture screen plates. These potential inducers are retested in a secondary screen to confirm that they were the organisms secreting molecules that alter bacterial gene expression in the reporter.
Although this method provides insight into the biofilm-forming interactions of Bacillus subtilis with soil organisms, it can also be applied to other systems, such as microbial interactions that affect virulence expression in Staphylococcus or Pseudomonas species. Indeed, it can theoretically be applied to identify microbial interactions that alter gene expression for any reporter in any genetically tractable microbe.
After generating fluorescent reporter constructs for your organism of choice, according to the text protocol, collect soil by removing the top 0.5 centimeters of exposed surface soil before using a spatula to collect soil in conical tubes. Add 0.85% sterile saline at a ratio of 10 milliliters per 1 gram of soil to make a soil slurry, and vortex it for 1 minute.